Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta, Emeritus Professor in the Department of Biochemical Engineering and Biotechnology at Indian Institute of Technology, Delhi. Today we are going to talk on module immunotolerance and immunosuppression to some extent from the paper Immunology. So, tolerance is the absence of the immune system to self-antigens. The early observations regarding tolerance were made with experimental animals. These observations helped the well-known immunologists Peter Medawar and Sir McFarlane Burnett in giving final shape to what we now have discussed or what is known as clonal selection theory. So the objectives of this module are to understand how innate immunity does not destroy own cells of the animal, to understand the phenomenon of central tolerance and peripheral tolerance. Finally, to understand that how neuroendocrine system controls the immune system. It means that the concepts basically involved are that the tolerance involves phagocytes and the two kinds of lymphocytes. This tolerance by these kind of cells, that's where the interface between the immune system and the neuroendocrine system is there. And we also see that how these two kinds of lymphocytes, B cells and T cells, have two broad categories of tolerance, broad classes of tolerance, what we call central tolerance and what we call peripheral tolerance. So one of the early dilemma facing biologists was that an immune system which reacts to a foreign antigen fails to do so against self-antigens present in the animal, in spite of the fact that the nature, essential chemical natures, structural nature of these self-antigens was not different in any way as compared to these foreign antigens. So, as Sir McFarlane Burnett said in 1976, quote, "For many years I have implied that the basic feature of immunity was the capacity to differentiate between self and non-self," unquote. What made this complex issue to understand that this capacity has to be present in both innate and acquired immune responses. The complete acceptance of the clonal selection theory requires that not only it should be able to explain the reaction to non-self, this feature must coexist or be balanced or regulated by lack of reaction to the self, lack of reaction to the self antigens. As in many other aspects of immunology, biologists were curious to understand whether tolerance is genetic or somatic. That is, it is part of genetic apparatus or it operates maybe at the protein expression level. As in the case of immune responses, tolerance, which is essentially lack of immune response, arises out of both. The cellular mechanisms are genetically controlled. Biologists who were convinced that genes decide everything were surprised that tolerance can be learned. The difficulties of transplanting organs were known quite early. In fact, so was the fact that these difficulties are not encountered at the embryo level in amphibians and chickens. In 1945, Ray Owen carried out serological studies with blood of non-identical bovine twins. These had mixture of blood types which persisted throughout the lifetime of twins. The puzzle was that if each calf was born separately, each will have single blood group type present in the mixture form of blood of twins. In that case, blood of X calf injected into calf Y would provoke immune response. In retrospect, this was the first clue that tolerance can be acquired either at the fetus level or very early in life. In rare instances, non-identical twins sharing the placental circulation in the uterus have different blood cells but tolerate cells from each other and accept mutual skin graft. In other cases, as shown in this slide, non-identical twins from different placentas do not acquire this tolerance. The inference was that the undifferentiated cells, either the stem cells or lymphocyte precursors, if they are mixed, these develop tolerance to each other indefinitely. Around 1957, we still did not know that there are two different kinds of lymphocytes. However, in that year, Sir Burnett suggested newly differentiated immunocytes were either destroyed or were made incapable of proliferation if exposed to an antigen. Some early experiments were carried out by Beatrice Mintz with the tetraparental mice, separately by Frank Dixon, Richard Smith, and William Weigel with rabbits, whereas Avrion Mitchison and David Dresser worked with mice. So each of the three groups were working with their individual and different systems. The observations of these investigators together provided valuable early insight into tolerance, and these are listed here. The picture which seems to emerge was as follows. Number 1, large and repeated doses of antigen can induce unresponsiveness in adults. It was felt that perhaps constant circulation of antigen is the criterion. Younger animals require small doses. When rabbits were injected bovine serum albumin at first week after birth, they became unresponsive. If the injection was given 3 weeks after birth, only 27% become unresponsive. Delaying injection to 4 weeks led to no unresponsive rabbits. Rabbits, however, when given a single large dose of BSA early in life, slowly regained response, but it was not restored completely. In adult mice, injection of soluble gamma globulin of human or bovine source showed that it has a long life and led to unresponsiveness. This persisted even after the antigen had disappeared from circulation. Interesting enough, aggregated gamma globulin, if injected, did not lead to unresponsiveness. It was also found that unresponsiveness could not be transferred to untreated animals by the transfer of serum or cells. GL Ada's lab at Melbourne, Australia was another important center where the research activity on the understanding of the immunotolerance took place. C.R. Parrish in that lab used chemically modified flagellin. Flagellin is a constituent of filaments on Salmonella and has been an important experimental antigen in that institution. The work with this experimental antigen was carried out while establishing clonal selection theory. Hence, flagellin was available in pure form both as monomer as a multimer. Albumin and gamma globulins were also available in pure form to be used as antigens. This happened because during World War II, blood became an important system and the US government funded blood protein fractionation research. The result was pioneering work of Edsel and Cohen, which actually laid the foundation of protein isolation and purification as we know it. Horse, man and bovine blood were the main systems chosen and albumin and gamma globulin fractions were available. This knowledge or the protocols generated during that work led to different laboratories purifying proteins of their interest. The main techniques used were limited to the precipitation by organic solvents, salts and metal ions. Chemical modification of proteins had been reported and a systematic study of that area was also initiated by Stein and Moore as a part of their project to sequence bovine pancreatic ribonuclease A. Ribonuclease A was the second protein and the first enzyme to be sequenced. The choice of RNase A was made because a meat canning industry in Philadelphia was offering samples of RNase A free to the scientists as their contribution to the US war efforts. All this made possible for this laboratory to have flagellin in the pure form and to be able to also form the various derivatives of flagellin. All this enabled Parrish to use various monomeric flagellin preparations. These preparations were acylated to a different degree. Acylation was carried out because it was found to reduce the capacity of the antigen to react with the standard preparation of its antibodies in vitro. Flagellin has also been a useful antigen as it has a good antigenicity, that is, even in small doses it produces enough antibodies. It also has a short circulatory lifetime. Parrish found out that an acylated flagellin, which still retained its capacity to precipitate the antibody in vitro to a high degree, however, was no longer an immunogen. When injected, it produced no antibodies. Curious enough, at the same time, its capacity to result in a delayed hypersensitivity increased. This property in fact remained quite well even in case of another flagellin derivative which has lost both immunogenicity as well as antibody precipitating ability. Parrish in fact carried out quite extensive studies with flagellin and its chemically modified derivatives. This particular slide, for example, produces some data which emerged out of these studies. This slide shows the production of tolerance by flagellin modified by cyanogen bromide. By that time, it was known that cyanogen bromide actually cleaves the protein at places where the methionine is present and accessible to the reagent. These studies indicated that there is a reciprocal relationship between cell mediated immunity and humoral response. It may be interesting to note two interesting facts. Firstly, these earlier workers, and so many of them got the Nobel Prize, had so few tools, yet they could infer so much out of their data with clear thinking and proper controls while designing the experiments. Secondly, many areas, especially at the early phase of their development, greatly profit from developments in other areas. The US government's investment in fractionation of the blood proteins as a part of their war efforts impacted so many areas in biochemistry or even biology. Immunology was one of these. People who fruitfully employed this knowledge included biologists. Lack of their mental insularity and openness of scientific communication was very important to the development of immunology in those days. Selection theory. Many leading workers led by Burnett had shifted focus on understanding tolerance. We will now see how the above experimental observation and a lot more which followed these were reconciled with the framework of clonal selection theory. Tolerance can be viewed as a part of regulation of the immune response. So let us first discuss regulation within the innate immunity system. Two broad ways it operates are lack of recognition of cells. This is called ignorance. During their life cycle, cells can change their surface structures. In that case, this ignorance vanishes. The second way the control of the innate immunity operates is by the presence of inhibitory structures on such non-immune cells. Phagocytes are important components of the innate immune system. So the first question is how these macrophages and neutrophils do not attempt phagocytosis of healthy cells. It may be recollected that the phagocytes recognize foreign cells of microbes via their pattern recognition receptors. Cells either do not have surface antigens recognized by these receptors. In many cases, these are covered up, for example, by sialic acid. In fact, this feature also is a part of how old erythrocytes and other dead cells are recognized by phagocytes. The old erythrocytes shed away sialic acid, exposing N-acetylglucosamine. This is recognized by PRR and phagocytosis commences. Similarly, with nucleated cells, death results in many surface molecules becoming more exposed. For example, phosphatidylserine is a phospholipid which is a part of the biomembrane, normally a part of the inner surface. Apoptosis results in phosphatidylserine flipping to the surface and being recognized by phagocytes. Natural killer cells kill virus-infected cells. Normal cells survive because of the balance between killer activation receptors called CAR and killer inhibitory receptors called KIR on the NK cells. KIR recognize MHC molecules on cells. In virus-infected cells, expression of MHC class I proteins is downregulated. This allows CAR to override and NK cells kill infected cells. The third key part of the innate immunity system is the complement pathway. The one which is antigen independent is the alternative pathway. You may recollect that C3 in that pathway is activated because some microbial surfaces provide stabilization of the hydrolytic activities. The cells in fact have inhibitory molecules on their surface membranes. This slide shows various regulatory proteins of the complement system and their function. As these regulatory proteins are expressed by cells and not by pathogens, complement thereby has the ability to distinguish between self and non-self cells. Action of some of these regulatory proteins can be seen here. MCP, for example, promotes C3b and C4b inactivation. CD59 prevents binding of C5b, C6 and C7 complexes to host cells. Now we can move over to the discussion on how adaptive immunity is regulated. So that tolerance is inbuilt feature of the acquired immunity. Most of the empirical and semi-empirical observation which we referred to earlier have been fairly clearly understood within the framework of clonal selection theory. Cells and T cells with specificities directed towards cells are deleted. This process is called clonal deletion. You may recollect that precursor T cells migrate from bone marrow to thymus wherein they develop into immunocompetent T cells. Those T cells which display high affinity to MHC class I and II antigens undergo apoptosis. Even out of these small percentage of T cells which survive, some still have affinity for self and exit thymus to migrate to peripheral lymphoid tissues. Similarly, clonal deletion of the B cells with potential to attack self molecules or cells takes place through apoptosis. The surviving cells leave fetal liver or bone marrow and reach peripheral or secondary lymphoid organs and tissues. Also, immature B cells having receptors specific to self antigens can become unresponsive or tolerant or allergic. These cells are not eliminated. We had discussed this during B cell development. This phenomenon involves editing of receptor specificity by light chain rearrangement and receiving incomplete activation signals, leading to downregulation of IgM while retaining IgD. Peripheral tolerance is necessary for achieving tolerance to self antigens which are not present in the primary lymphoid organs or reach there by bloodstream. Naive T cells require signal via TCR as well as from co-stimulatory molecules. CD80 and CD86 are essential co-stimulatory molecules on APCs. CD86 interacts with CD28 on T cells. APCs also release cytokines. Unless APCs are bound to the microbes, upregulation of CD80, CD86 does not take place. So by default, MHC self antigens interact with naive T cells, resulting in an energy. As a fail-safe rearrangement, if any T cell activation does take place, these express CTLA4, which interacts with CD80 much more strongly than CD28. Binding of CTLA4 to CD80 sends a negative signal to T cell, which inhibits their activity. B cells require triggering through their CD40 molecules to progress through activation and maturation. Interaction of CD28 on T cells with B7 on B cells is necessary to induce expression of CD154. This binds to CD40 on the B cell, acting as the second signal for activation. Corresponding TH cells have been already eliminated during thymic education, during central tolerance. In the absence of this cooperation, B cells become energic. Antigens require T cell cooperation to respond to T cell dependent antigens. Most of the autoantigens are present at too low level to become target for T cell recognition. Those which generate cell peptides at a level high enough to be recognized by effector T cells, but low enough not to induce tolerance. Even in such cases, T cells fail to get activated because of the low affinity. This phenomenon is called T cell ignorance. During the neonatal period, tolerance is established to self antigens. B cells and T cells have receptors which differ in their specificities. For example, B cell receptors are immunoglobulins. All these have common Fc portion and collectively are present at high concentration to provoke tolerance. The highly variable sequences which define antigen specificity are determinants is present in too small a concentration to provoke tolerance. While discussing Jernet's network theory, we had learned that Jernet called these idiotopes, and antibodies directed against idiotopes were termed anti-idiotypic antibodies. So both T cell receptors and B cell receptors will generate these anti-idiotypic antibodies. The administration of anti-idiotypes at neonatal stage has been found to lead to even lifelong effects. T15 is a major idiotype in the antibody against phosphatidylcholine. If anti-idiotype T15 antibody is administered to a neonate, long-lasting suppression of antibody production against phosphatidylcholine is observed. Significantly, doing the same with adult has limited effect. Hence, while Jernet's network theory may not have been successful in explaining all features of the immune system, the concept of idiotypes or idiotopes given by him was valuable in understanding tolerance. The in vitro experiment suggests that idiotypic antibody idiotype network may operate in inducing tolerance. The neuroendocrine regulation also plays an important role. The main role of the nervous system seems to be dampening down of inflammatory responses and maintaining homeostasis via negative feedback loops. The hypothalamus pituitary adrenal axis releases glucocorticoids, which are hormones secreted by the adrenal cortex. These suppress inflammation by inhibiting release of cytokines by dendritic cells and macrophages. Antigen presentation and mast cell functions are also inhibited. These multitasker molecules, that is glucocorticoids, also influence trafficking of the immune cells. Dual mechanism of decreasing the chemokine production and altering the expression of their housing molecules seem to operate in this cell trafficking. To summarize the lecture, we learned that tolerance is acquired at the fetal or neonate level by animals. Innate immunity also involves tolerance. Acquired immunity involves central tolerance and peripheral tolerance. We learned that the immune responses are also subjected to neuroendocrine regulation. Tolerance and immune suppression are twin phenomena. We had earlier seen the role played by T regulatory cells in regulation of the immune response. In autoimmunity module, we'll see how these mechanisms fail. In fact, because of these sophisticated tolerance mechanisms, there are fewer autoimmune diseases. Thank you.